So in this video, I will explain you about something about virus multiplication. So you can see here the orange peg heading of virus multiplication. So I will explain you to using diagrams and in a short manner in such a way that you can easily understand. So there are many steps of virus multiplication. That is actually virus. We are talking right now about reproduction of virus. That is, this is nothing reproduction by a virus. So I hope you might know about virus that they are considered to be as the border line between living and non-living organisms, and they have somewhat slight structure in the color. And then, so I hope it is in that diagram. Actually, there are many other different types of structure, but this is a typically drawn virus structure. So this is a virus. It is it is a really it's a really very harmful kind of thing we can call, because it can even infect human cells as well as the bacterial cells. So in this way, we will see how a virus multiplies itself in an host. So for this, we will take a host as a bacteria. Let's take some random bacteria. So this is my bacteria. So here is this nucleus, you can call, and then this is all the cytoplasm. I'm not going to draw the rest of the organelles, that because we are now focusing on virus multiplication. So this is what this is my bacteria. So this is now we are talking about stage one, where the virus attaches itself to this cell or to the host cell with this leg-like structure. You can see here they are these are known as tail fibrils. So let me hold those in. These are tail fibrils. Tail. So it attaches itself with these tail fibrils, and even there are some smaller structures known as. And this process is known as adsorption. So adsorption, adsorption. So here the virus. Let me draw the virus here. So this is my virus. This color is the protein material, and in this way it has attached attached to the bacterial cell, and this is the first step, which thus it is now the process of infecting it. So let me first change the color of the arrow, seems more colorful, more better. And then we go to the second stage, which is injection. First, I forgot to do a very important thing, that is I forgot to show this DNA material, that is what all the functionality and virus. So the second step is the injection step. So let me copy my bacteria to save you and my precious time. So let me copy my bacteria, and this is my bacteria. And now it's going to, as the name suggests, it's going to inject its DNA material, or it can be an RNA material, into this host. So like thing. So again, let me copy my virus also. Let me copy that. It will go a little down here. Yes, this is the back. Oops, cut it. Some problems. Okay, no worry. So it it's to be injecting some that was present earlier into the virus. It's DNA material, so or you can say it is the injected nucleic acid, or because there can be RNA as well as DNA. So it is injected nucleic acid. Yep. This, this phase is called as injection. It was adsorption, not absorption, it was adsorption. And this is injection. Injection. And now, let me make some more space. Now it will go into the third stage of DNA replication. So it will replicate its DNA. That is, now let's assume that this virus that I have drawn, a very bad virus that I have drawn, is contains a DNA material. So I will not talk it as a DNA material. So so, so it's now going to replicate this material. Just that it's going to go into replication now. So let's see now. Let me again copy my bacteria now. Now the outer body, now the outer body of that virus, the yellow color that I have drawn here, is no more useful as all the actually we can call it even a dead because all the necessary thing that is the nucleic acid that used to control it are inside the bacteria only. So or you can call it the host cell only. So this is the replication killing the it's killing the bacteria's nucleic acid, or it's killing the bacteria's DNA and is copying its own DNA. It happens at a way faster way. Happens at a really very fast way. So this is what we call it as a DNA replication. Uh, like that. So this is the third step that has happened in the process, which is replication replication of DNA. And now let's jump to fourth step. Now let's jump to the fourth step of protein synthesis. Now these the replicated DNA. Now these replicated forms the necessary structure in the host itself. That is the necessary structure is this. That is this. You can see like the head and the leg-like or tail-like structures. It will be formed in the host cell itself. So when I call my bacteria here, it is now in serious trouble actually. So it's a little difficult for me to show. Can't call it material, but can even call it as for simple language. We can call it as necessary organelles or parts. And then this is uh, so this is the protein synthesis. So the protein synthesis is taking place here. Thus making thus it's the formation of necessary material. This is my protein synthesis. Now the fifth step, it's the second last step now. So the fifth, so now the fifth step is assembly. Now this is this is not a fully grown virus right now. That is, there are some proteins that are need to be synthesized. So these all this stuff is taking place here, and then the next step is of assembly. So again, now I will copy this image. So it's now going to assemble itself. So let me draw it more neater right now because it's now at its best stage. It's now grown totally. So it has got everything now. It has got its tail. It has got its tail pins. It has got everything. Tail fibers. And so this is the This is the assembly stage. This is called as assembly. Oops, called as assembly stage. And the last stage is release. Now it has now completely grown, and it will say that, hey, I will not stay for a longer time here. Instead, I will go out uh, and infect some other cells that can get it to be infected. So it's you can call it as it's not at all a stable thing. So it will now break open the bacterial cell. Let this. So it will now break over, break, break up that bacterial cell. It is now already decomposed, or you can say killed everything in that bacterial cell, and now it's going to release. And it's not the one that is not going to release. There are almost 200 or 300 of them that are going to release from this single thing. So what miracle type of thing. It's such a small thing, the small virus has done. So let me draw this in this way. There are many viruses form, and these are not at all in the baby stage or something. These are fully grown, and that is they are ready to replicate. Oops, they're ready to carry out this further process of virus multiplication again. So, so my drawing is dangerous on computer. In this way. And this is the important thing. Never forget to draw the nucleic acids or the DNA material here. And in this way, it has replicated, and now, uh, and now it will, infect some other bacterial cells. So the cells by the side that are were uninfected. So this even happens in your human cells also. So maybe call this as viral infection or such things takes contains place. So this will go here. It's just a imagination type of fragment. So this will go here, this will go here, this will go here, and then this will again, each of them will again go back to that first stage, and it will keep on, doing all this till the host is not actually dead. So we take a lot of medicines and some necessary things for viruses. So this was nothing but virus multiplication, a little hard topic, but I hope you might have understood this. Thank you.